My name is Cameron Bunce, and my group discussed the effects of pH level on trypsin. So a little background about trypsin. Trypsin is a digestive enzyme that is produced naturally in the pancreas. Trypsin can speed up the hydrolysis of the peptide bonds, hence breaking down the proteins quicker so they can be absorbed by the human body for energy and nutrients. It speeds up chemical reactions because it is a catalyst for hydrolysis. This means that they increase the rate of hydrolysis. Enzymes are needed for the metabolic pathways in the body, respiration, and digestion. When enzymes function properly, homeostasis is maintained. Trypsin is a protease that works to speed up the breaking down of molecules into protein structures such as peptide bonds and amino acids. Trypsin is crucial for the digestion of proteins in the small intestine. The activity of trypsin varies depending on the pH of the solution. So my group analyzed the effect of pH on enzyme activity. So we hypothesized that trypsin would have the highest optimal enzyme activity level at a pH of 9, and it will begin to decrease as the basicity of the solution increases. And our rationale for this is that based on our own data and other groups in the class, we seemed like everyone was achieving an optimal at a pH of 9. So our methods and materials for this lab, our substrate used was azocasein, which is prepared chemically by adding sulfamilinide groups to the milk protein casein. Our experimental variable conditions during the experiment were the pH levels of 7.5, 8, 8.5, 9, 9.5, and 10. And the control variables in the experiment were the salt concentration of 0.15 molarity and the temperature of 37 degrees Celsius. We used 3 different replicates during our experiment, and we also used a negative control in order to confirm that the results were actually due to the pH of the solution and not any other outside factors. Our negative control solution was heated to 100 degrees Celsius in order to denature the enzyme trypsin and stop the reaction completely. The procedure we followed during the experiment started with adding 450 microliters of reaction buffer, or the pH, 450 microliters of azocasein, and 50 microliters of trypsin to a centrifuge tube. We then incubated the tubes and their replicates in the hot water bath for 10 minutes at 37 degrees Celsius, and we did this to increase the enzyme activity and lower the activation energy for the reaction. We chose 37 degrees Celsius because that's the optimal temperature for enzyme activity in the small intestine of the human body. So after we removed the tubes from the bath, we then added 500 microliters of TCA to stop the reaction completely, and then we centrifuged the tubes at maximum speed of 14,000 RPM for 2 minutes to ensure that the TCA was evenly mixed within the entire solution. We then transferred our centrifuge tubes to centrifuge solutions into cuvettes. The cuvettes are tubes that fit into the spectrophotometer and allow us to run absorbance tests. The final step is then to test each pH cuvette and its replicate's absorbance rate at 440 nanometers. The spectrophotometer allows us to determine which pH had a higher average absorbance rate, and the absorbance rate directly corresponds with enzyme activity, allowing us to make a determination of which pH would be optimal for enzyme activity. To blank the spectrophotometer, we filled one of the cuvettes with deionized water, and that sets it to 0. So our results for this experiment was that we actually ended up getting an optimal pH of 10, as you can see, as that's the highest when it comes to the absorbance rate. But we're not exactly sure if our data is statistically significant because there's high variability in our data, and after calculating the p-value and standard deviation, it shows very high variability in our data, leading us to conclude that something must have gone wrong during the experiment. So We believe that our experiment was adequate to test the effects of pH on enzyme activity. However, the negative control in our experiment was potentially flawed due to the fact that the negative control tube was not placed in the 100 degrees Celsius hot water bath at the same time that our other trials were placed into their warm water bath. So this gave the other trials in the warm water bath extra time to react before the negative controls did. And due to this, the experimental tubes had reacted longer than they were supposed to, because we waited to add the TCA until after the negative control was done incubating in the hot water bath. So the delay in the addition of the TCA in the experimental tubes potentially caused error and high variability in our data. Another source of error in our experiment could have occurred during the mixing of the pH solutions. So because we used increments with our solutions, we're not 100% sure if we were actually at a pH of 7.5 or 8.5. It could have been a little higher, a little lower based on human error when it comes to measuring. But in conclusion, we hypothesized that the optimal pH level for enzyme activity would be at a pH of 9 based on our own and other groups' results who also achieved that optimal activity with a pH of 9. However, our hypothesis was rejected by our experiment after we analyzed our results. Our data showed that the optimal pH level for enzyme activity occurs at a pH of 10. Our results in our experiment are not statistically significant due to very high standard deviations and p-values, proving a high amount of variability.